Good morning, Honorable. I'm Anjali Krishna, currently doing PhD first year under the supervision of Dr. Krishna and co-supervisor Dr. Renuka Surajala. So my topic is inferring variants of unknown significance in rare disease genetic. It's an all Indian centric studies. So first of all, I'm taking the rare disorders. So first question is, are rare diseases are rare or not? So based upon the literature review, you can see in US 2 lakh people are already affected some kind of rare disorders, and 1 per 10,000 in Taiwan. If we take an India had 85 million population, or 76 to 85 million population. So almost approximately 7.8% populations are tagged as a rare. So mostly rare disorders are usually monogenic and largely heterozygous, and the important factor is allele frequency. It's less than 0.01 for rare disorder, and some extremely case is less than 0.001. And it have many uncharacterized mutations, and these mutations are largely variants of unknown significance. So the parallel genotypes are not necessarily transcended to the children, which means there are two types of mutation. It could be rare or de novo mutations. And rare disease variants in congenital pouch colon and anorectal malformations anomaly were discovered. That means like we have two samples like congenital pouch colon and anorectal malformation. So the congenital pouch colons is a condition of the shortened and pouch dilated colons, and they usually associated with the anorectal malformation. And the pathogenesis of the embryology of congenital pouch colons are not well understood, but dietary environmental factors that may be affected or contributing factors in this pathology. Then it's mostly visible in India than the other countries, with the reviewer showed like 90.2%. And here some of the papers are give us the references. So here you can refer like 4 papers which elaborate the case study. And our lab has also worked the inferring rare variants in aplastic anemia, aquatic aplastic anemia. The main symptoms are fatigue, fever, headache, dizziness, and shortness of breath, unexplained of easy bruising, rapid or irregular heart rate, etc. So the important thing is the missing link between the rare diseases. So rare diseases are like maybe it leads to autoimmune disorder or infectious diseases, neurological disorders, or risk factor or prevention. So if we find that missing link between these diseases, we can easily provide the treatment, or we can easily find some kind of treatment or treatment kit or what. And the problem statement in this problem, like the thing is like growing myths and challenges that may be that many rare disorders are autosomal dominant or recessive. So knowledge on variants of unknown significance and lack of the consensus and genetic test in detection of rare disease. So does rare disease have preferences for any specific allele or not? So these are the research gaps or the problem statement which we have to solve. So my main objectives, based upon these, my main objectives are kind of like to check whether or not genetic variation is best seen in non-coding RNAs, or to identify the role of variants of unknown significance in rare diseases and establish causal relationship between variants in the genetic of rare disease common to India, to identify the genetic susceptibility of variant of unknown significance, finding unique roles in their pathogenesis. And the last one is to dock the variant of unknown significance emerging from the rare phenotypes with targeted ligands and established causal relationship with disease causal genes. And here I upload the PRISMA guidelines. Here you can see. And coming to the expected outcomes, so establishing causal relationship for non-coding RNAs and identifying genetic variations in them in some selected rare disease of India, and developing a standard operating procedure or framework for rare disease and India context, taking variant of unknown significance and attempting to understand long range sequencing chemistry using OND-median for identifying rare exomic variants and finding the roles of interfacial or causal interfacial, that means the causal variants residues across the rare disease. That's all. Thank you.